Welcome to Kognito Key. Today we're going to unlock the science behind a really fascinating biological puzzle.
So let me ask you something. How would you save a plant from a virus that has absolutely no cure? Or what if you wanted to create 1,000 perfect copies of a super rare flower? It sounds like science fiction, right? Well, the answer lies in this amazing biological technique, and we're about to dive into exactly how it all works.
Okay, so the solution is a technique called Meristem culture, and it all starts with this tiny, almost magical part of the plant. The secret, it's hidden right there, at the very tip of a growing shoot.
So, what is this thing exactly? Well, Meristem culture is a special kind of tissue culture. Now, tissue culture just means growing plant cells in a lab, in a petri dish. It uses a process called vegetative propagation, which is really just a fancy word for cloning, to create plants that are genetically identical. It's a pretty brilliant method, actually, first developed way back in 1952 by two scientists, Morel and Martin.
So let's get to the heart of it. What is the meristem? You can think of it as the plant's main control center for growth, or maybe like its own personal fountain of youth. It's this tiny little dome of cells right at the tip of a shoot or a root, and these cells are just constantly dividing. But here's the really cool part, the part that makes this whole thing possible. It's naturally pathogen-free. So even if the rest of the plant is sick with a virus, that virus just can't seem to reach the super fast-growing tip. It's like this clean, protected little bubble. And when I say tiny, I really mean tiny. We're talking about a section that is just 0.1 millimeters wide. To put that in perspective, that's about the thickness of a single strand of your hair. Can you just imagine the incredible skill and precision that takes to work with something so small?
Okay, so you're probably wondering how this is actually done. Let's go inside the lab and I'll walk you through the process step by step. First up, you start by taking a small shoot tip from the plant. This little piece then gets a really thorough cleaning. You've got to get rid of any stray contaminants. Then comes the really delicate work. Under a powerful microscope, a scientist has to carefully dissect away the outer leaves to expose that tiny, pristine meristem dome. This precious little speck is then placed onto a special nutrient shell. Think of it like a complete balanced meal to help it grow. It's kept warm and cozy, and eventually it develops into a brand new, tiny shoot. And finally, this little plantlet is ready to be moved into a pot with some compost and gently introduced to the big, wide world of the greenhouse.
It's a really fascinating process, right? But why would anyone go through all this trouble? Let's take a look at why this technique is so unbelievably important in the real world. Oh, the impact is absolutely huge. First and foremost, it lets us create completely virus-free plants from a parent plant that was actually infected. It's incredible. But it's also used for rapid multiplication, what scientists call micropropagation, letting you make thousands of clones from just a single plant. This is so important for breeding new hybrids, for conserving the genes of endangered species, and even for making it easier to transport plants across international borders, since they're certified as being disease-free.
Of course, no technique is perfect. There's always a balanced view. On the plus side, it's amazing. You get virus-free plants and a fantastic way to conserve plant genetics. The meristem even has its own built-in growth hormones that give it a little boost. But there is a downside. The process itself is incredibly difficult. Isolating that tiny little tip requires immense skill, and it can take a very long time for that tiny speck to grow into a full-size plant.
This quote really just sums it all up perfectly. Meristem culture isn't just some cool lab experiment. No, it's a really powerful tool that has a huge impact on our food supply and on conserving the planet's biodiversity.
And all of this leaves us with a really fascinating question for the future, doesn't it? We now have the technology to create perfect, healthy copies of almost any plant we want. So, what should we focus on next? Should we be rescuing more endangered species or creating crops that are more resilient to climate change? The possibilities are truly endless.
So, if you found this little explanation helpful and interesting, please do subscribe to Kognito Key. And don't forget to like this and share it with others who you think might be curious too. Thanks so much for watching.